everybody, welcome to BioLibrary. And after collagen and elastin, let's discuss about keratin, which is again a protein which is found in your hairs, nails, and जो animals होते हैं in their scales, horns, wool, nails, feathers. So keratin protein is found by all vertebrates. First point. Chief structural component of hair, scales, horn, wool, nails, and feathers. I've told you that it is a universal rule that fibrous proteins are structural protein, whereas globular proteins are functional proteins. Okay, so keratin is present in two forms. First of all, keratin is a protein of intermediate protein family. So you know there are three categories in this: microtubules, microfilaments, and in between them intermediate proteins. Okay, so it comes from intermediate protein filament family. Two forms. Alpha forms is found in mammals and beta is in reptiles and birds. So it is found in all the vertebrates. Out of vertebrates, alpha form is found in mammals and beta form is found in reptiles and birds. So let's talk about what is found in mammals. So alpha, because alpha helix is present and the amino acids which are present there are single digit codes. I have represented ALVIMF. So what you can do, I think you all know what is the extension for this. That is alanine. This is leucine, this is valine, isoleucine, methionine, and phenylalanine. Okay, so there are disulfide bonds present. This is disulfide bonds are present in vertebrates, also in few invertebrates. So alpha type is also present in few of the invertebrates. Now let's talk about beta type. Beta type again is present in reptiles and birds, and here you have beta pleated sheet structure. So these initials, they come from alpha for alpha helix, beta for pleated sheet structure. So the arrangement is antiparallel arrangement of beta strands. Again, disulfide bonds are also present here as well in beta form. So two types, alpha and beta form. Alpha in mammals because alpha helix is present, beta present because of beta pleated sheet-like structure in reptiles and birds. So this is the structure of alpha keratin polypeptide as a whole. If you see, there can be acidic amino acids in it or they can be neutral or basic. Depending on that, type 1 alpha keratin polypeptide has acidic amino acid, whereas type 2 contains neutral or basic amino acids. Let's talk कि इसकी complete synthesis as well as release किस form में होता है. So you may have two type 1 combining or you may have type 1 as well as type 2 combining together. Okay, so type 1 and type 2, if they are of two different type, that is one is acidic type and the other is either neutral or basic type, then it becomes what? A heterodimer, because both of the mers, both of the subunits are different. So heterodimer, which is parallel, this is a sign for parallel spin. It is a polar structure because it will have directionality. Okay, now two heterodimers. First, we formed a heterodimer. Now, two such heterodimers, for example, this is one and the other one as this. Two heterodimers, they combine together and they'll form a structure which is a tetramer because it is combining two dimers. Two dimers means 2 plus 2. This is a dimer, this is a dimer. It makes 4, that is tetramer. So it's a protofilament. It's called a protofilament which is used as a unit of your alpha keratin, such as protocollagen, protoelastin. Similarly, you have protofilament. It's a tetramer. Here it's a soluble structure. So right now, till now, you can't say that it's a fibrous protein. So till now, it's a globular protein. It can behave like a globular protein. Okay. Now these two protofilaments, when they combine together as in octamer state, they will make one protofibril. Two protofilaments combine together to form one protofibril. Now such four protofibrils, say for example this is one protofibril, such four protofibrils will combine together to form one microfibril. So this is what? This is a microfibril. Now so many microfibrils, they will combine and form a macrofibril, which is non-soluble and which is released in the form of alpha keratin. So that's the journey of alpha keratin from synthesis. Let's talk or revise it again. You have two subunits. You have subunits which can contain acidic or neutral or basic amino acid. Suppose you have one acidic, you have one basic amino acid containing chain. They combine together, you form a heterodimer. Then from a heterodimer, two heterodimers combine and you form a protofilament. Such two protofilaments combine and you form one protofibril. Such four protofibrils combine together, you form one microfibril. Such microfibrils combine together and you form macrofibril, non-soluble form, and it is also known as alpha keratin. And here your keratin topic ends. I have dealt with collagen, elastin and keratin today. So I'll take your quiz on Telegram at 8 p.m. tonight only. You will get 30 seconds for each question. Let's see how many do you score. The complete marks for your test will be 45 marks. That is, each question contains 3 marks and 1 marks. You can neglect 1 mark for your wrong answer. Thank you everybody and keep studying.